Okay, today kids we're going to talk about mitosis, which is also called the cell cycle, and how cells divide. Phases of the cell cycle consist of interphase, which also consists of three different phases within itself, and the mitotic phase, which is called mitosis, or the actual dividing of the nucleus. Interphase is broken down into three categories. G1, which is the growth, where cells will actually start to grow, and then it will enter the S phase, which is the DNA synthesis, where DNA is duplicated, because like our human cells, our somatic cells in humans contain 46 chromosomes. Well, we'll actually divide or multiply that into 92, and then it enters the growth 2 phase, which checks on and makes sure that the DNA was copied correctly. And after the cell goes through this process, then it can enter into the mitosis phase, or dividing of the nucleus. Here are some pictures and functions of cell division, reproduction. Here's an amoeba that's actually being divided here, and then this micrograph shows a sand dollar embryo shortly after the fertilized egg is divided, and then tissue renewal like in your bone marrow cells. And cell division is an integral part of the cell cycle, results in genetically identical daughter cells. Cells duplicate their genetic material before they divide, ensuring that each daughter cell receives an exact copy of the genetic material, DNA. Now, this is also don't get mitosis and meiosis confused. Meiosis, you're going to get genetically different cells than you would in a mitotic division. Genetic information is carried to genome. You know, we talk about and discuss what our genome is. Our genome is just all the genes that make up, our body, or our animals, or all plants, and they're packaged into chromosomes. An average eukaryotic cell has about 1,000 times more DNA than an average prokaryotic cell. Now, the difference between a eukaryotic cell is, like humans, everything that consists of in the kingdoms of the animal, plant, fungi, and protists. These are all eukaryotic cells, and then the prokaryotes are like your bacteria and archaeobacteria. The DNA in a eukaryotic cell is organized into several linear chromosomes, whose organization is much more complex than single circular DNA molecule in a prokaryotic cell. And a prokaryotic cell, like I said, is a bacterial cell, which their DNA will be in a single loop, where ours is linear. All eukaryotic cells store genetic information in chromosomes. Most eukaryotic cells have between 10 and 50 chromosomes in their body cells. Human cells, such as like ours, have 46, and they're paired up, so you have 23 nearly identical pairs. Chromosomes are composed of a complex of DNA and a protein called chromatin that condenses during cell division. DNA exists as a single long double-stranded fiber extending chromosomes' entire length. Each unduplicated chromosome contains one DNA molecule, which may be several inches long. In the human body, one cell, and if you was to string all the DNA together in just one cell of the human body, it would roughly be over 6 feet long. That's how much DNA that the body has. Every 200 nucleotide pairs, the DNA wraps twice around a group of 8 histone proteins to form a nucleosome. Higher order coiling and supercoiling also helps condense and package the chromatin inside the nucleus. And this is the only way that basically DNA can end up in a tiny, tiny cell, is that this tightly constricted of DNA around those histones are tightly wrapped. Karyotype is what you would go and to a doctor and see if that if you had something wrong with your chromosome, but is a visual representation of the chromosome in a cell. And every cell in your body, other than a sex cell, contains those 23 pairs of chromosomes. In humans, each somatic cell has 46 chromosomes made up of 2 sets. One set of chromosomes comes from each parent. So you inherit one of those homologous chromosomes from dad and one from mom. Non-homologous chromosomes look different, control different traits. Sex chromosomes are distinct from each other, and characteristics are represented as X and Y. If you're an XY, you're a male, and if you're an XX, you're a female. In a diploid cell, The chromosome occurs in pairs. That means like any somatic cell or body cell other than a sex cell is a diploid cell, and then the sex cells referred to as haploid because they contain half the number of chromosomes. A diploid has 2 sets of each of its chromosomes. A human has 46, so 2n, 23 pair equals 46. In a cell in which DNA synthesis has occurred, all the chromosomes are duplicated and thus consist of 2 identical sister chromatids. Homologous chromosomes look the same, control the same traits, may code for different forms of each trait. And what this means is that when you inherit these, when you get mom's 23, dad's 23, and they go to match up, they will match up 1 to 1, 2 to 2, and so on down to even the sex chromosomes, the X and the Y, or the X and the X. So you're getting each trait from each parent, so they may be just a different form of that trait. In preparation for cell division, DNA is duplicated and the chromosomes condense. Each duplicated chromosome has 2 sister chromatids which separate during cell division. So if you look on the diagram there, there's a linear chromosome, and you see the sister chromatids there, and then they match up right there in the middle at the centromere, and they will actually end up splitting when they end up splitting after metaphase. So because of duplication, each condensed chromosome consists of 2 identical chromatids joined by a centromere. Each duplicated chromosome contains 2 identical DNA molecules, 1 each chromatids, and we call those sister chromatids on each end. The centromere is a constricted region of the chromosome containing a specific DNA sequence to which is bound 2 discs of protein called kinetochores. They serve as a point of attachment for microtubules to move the chromosome during cell division. And that centromere will actually line up in the middle during the phase of mitosis in nuclear division. Diploid is cell possessing 2 copies of each chromosome. Homologous chromosomes are made up of sister chromatids joined at the centromere. Haploid, like your sex cells, just contain half the number of chromosomes. Phases of the cell cycle. As I mentioned earlier, the G1 is where the growth is. The S is when the DNA is actually duplicated. And then G2, you have your secondary growth or your checkpoint to make sure that the DNA was copied correctly. This is where a lot of cells will spend most of their lifetime is in the interphase stage, especially like your nerve cells, because it takes a long time for those to regenerate. And then some of them, the quicker ones, like skin cells, when you lose skin cells every day, you know, they're multiplying, so they will not stay in this phase for very long. As I explained before, the S, each chromosome replicates, producing twice the amount of DNA, and then the G2 checks it, and then we start mitosis. Each new cell receives 1 copy of every chromosome that was present in the original cell. It produces 2 new cells that are both genetically identical to the original cell. So if you have, during mitosis, you're going to produce a genetically identical 2 daughter cells from 1 cell. There it shows you some pictures of the G2 of interphase, and then the prophase. You will start to see when that happens that the chromatin will start to condense instead of looking like string inside of a ball there in the nucleus. And then it goes to prometaphase, where you will see the chromatins start to head toward the center. And then here at metaphase, you'll see them lining up in the center. And then anaphase, you will see each chromosome pair start to split. When they start to split, that means each one will go to the poles. And then in telophase, you will actually see them, the nuclear membrane start to reappear, and the chromosomes at the end. And then you will, in an animal cell, you will start to see what they call a cleavage furrow, to where the cells are going to split apart, forming 2 new daughter cells. And here is just some more definitive things of prophase that you could read, and metaphase where they line up in the middle, and the spindle, which will help pull the chromosomes apart. And you can see the spindle there. And then anaphase, the actual splitting apart of the chromosomes and going toward the ends. And then you got two like daughter cells at the end of the process. Now at the end of telophase, you're going to start to see what they call cytokinesis, which is splitting of the cytoplasm and all the other organelles. That way each cell will get the same amount of cytoplasm and organelles where the cells can function. And the difference between an animal cell, as you can see in the picture at the top and the bottom, you will see the cleavage furrow start to look like it's pinching apart, and the plant cells would actually start to form a cell plate, and you will start to see that form between the two cells. And you can see that in those pictures there. And there's some, probably that is a picture of some onion cells, which are good on the onion root tips there, that you can see all different stages of whether it's prophase, metaphase, anaphase, or telophase, and even cytokinesis there. And then you would actually start to see a cell plate. All right, and thank you.